Day 11, and the human was counting. I had the feed open on my secondary display. Cell 14, isolation wing, standard sensory protocol. And I watched the subject sit cross-legged at the center of the floor, eyes closed, lips moving in a rhythm I could not quite parse. No vocalization above threshold, no visible distress markers. This all analog within acceptable range, respiration. Slow, deliberate, almost meditative. 11 days of total isolation, no light variation. No sound input, no contact, no tactile surface complexity beyond smooth composite flooring and bare walls. We had stripped the cell to nothing because that is the protocol. And the protocol exists because 11 days is the number. Across 214 catalogued sapient species, 11 days is when the mind begins to come apart. Precise mechanisms vary by neurology. Some species dissociate, some produce hallucinatory output, some simply stop moving and wait to die. But 11 days is the wall. We have centuries of data. The wall does not move. I called up the Behavioral Dissolution Index on my primary screen and watched the human score update in real time. It sat at 0.4, baseline noise. I refreshed it twice, which I will admit was not a clinical response. Junior analyst Thavari leaned over from the adjacent station. Day 11, she said. We should be seeing early fragmentation. I am aware, I said. The score, I see the score. She did not say anything further. Neither did I. We watched the feed together for another 40 minutes, and the human counted. On day 14, it began scratching the wall. Not aggressively, not randomly. It used the edge of a meal tray corner. We had left the tray, as per standard protocol, and made small, precise marks near the base of the eastern wall, in a row. One mark per standard unit of approximately 26 hours, which I did not understand at the time, because a human isolated from all light cycles should have no reliable mechanism for tracking the passage of days. When I raised this with my team, analyst Thavari ran the feedback against the environmental logs and found it anyway, the meal delivery interval. We deliver on a precise schedule. The human had inferred it from the cadence of the tray slot opening and was using it as a clock. I filed a note in the observation log. Subject demonstrated temporal inference from minimal environmental cues. Behavioral Dissolution Index. It was still noise. Less comfortable noise. Director, Vela Maris visited the observation bay on day 19. She reviewed the accumulated readouts without comment. Fingers moving through the summary display in the unhurried way she has when the data is telling her something she did not come here to be told. When she reached the behavioral dissolution scores, she stopped. These are the correct readings, she asked. They are, I said. Calibration verified. 3 times. She studied the chart for a long moment. The human's score had risen to 0.11 by day 17, which in any other subject would have been the leading edge of fragmentation. The first, hairline crack before the wall falls. In this subject, it had plateaued there for 2 days and then dropped back to 0.9. It stabilized, Vela Maris said. It reorganized, I said. Whatever internal structure it is using to maintain coherence, it appears to be self-reinforcing rather than depleting. She looked at me then, and I recognized the expression from 40 years of research. Not fear, not precisely, something adjacent. The thing that lives in the space between an anomalous result and its implications. Continue monitoring, she said, and left. By day 23, the wall notations had expanded. Alongside the daily marks, 23 now, 4 neat groups. The human had begun a second set of symbols below the first row. Smaller, more complex. Thavari's first interpretation was language. My first interpretation was language. We were both wrong. On day 26, when the pattern had grown enough to analyze, our translation module identified it as mathematical notation. Original notation, a symbol set the human appeared to be inventing in real time. Building from scratch the way you might build a scaffold before a structure worth supporting exists. I sat with that finding for some time. On day 28, I quietly requested a secondary calibration of all monitoring systems in isolation wing. Standard procedure when data is unexpected. The technician ran the full check and returned a clean bill. No sensor drift, no recording artifacts, no interpolation errors. The notation was real, the schedule was real. The dissolution scores were real. I thanked him and closed the maintenance ticket, and did not tell him why I had filed it. There is a category in our behavioral taxonomy for subjects who produce output during isolation. It is a late-stage dissociation marker, the mind generating internal signal to substitute for the absent external world. Schematic productions, repetitive drawings, phantom communications. We see it in species that fail at day 30, day 40. It is a sign of the wall beginning to give. This human's notation was not schematic. It was not repetitive. Each day's additions built on the previous days. It was developing. On day 31, Dev ran the notation sequence through our mathematical analysis suite and came back to me looking slightly pale. For a Vithrani means the inner eyelid has dropped one position. It's working on prime enumeration, she said. Starting from what base? Its own. It established the base first. The first 9 days of notation were the axioms. I pulled up the wall image from day 9. 9 days of careful scratch marks. I had classified as early-stage symbolic coping behavior. The human had been building a formal system. It had simply not gotten to the theorems yet. I updated the behavioral dissolution index entry for day 31 and then sat for a while longer than I should have, looking at the feed. The human was doing what it always did at this hour. What we had come to understand was its designated rest, period, one of 4 structured intervals it maintained across the day, with no external enforcement, no signal, nothing but its own internalized schedule. It lay on its back, hands folded on its sternum, eyes open looking at the ceiling. Not staring, looking. The distinction is not trivial. Staring is the dissolution response, the gaze that has stopped tracking, because tracking has become too costly. Looking is active. The human was still in there, still choosing. Week 7, day 48. I presented the accumulated findings to Director Velamaris and the full review board. 22 slides, 14 data series. The behavioral dissolution scores, which had never exceeded 6.13 and were currently sitting at 6.18, lower than day 3. The notation wall, which now covered the lower third of the eastern surface and part of the southern. The voluntary schedule, documented across 48 continuous days without a single structural collapse. The temporal inference. The mathematical construction. I also included, on the final slide, a single comparison. The mean dissolution score at day 48 for the 37 other species we had observed in isolation protocol studies over the past century. The chart was not complicated. 36 lines intersected the critical threshold between day 8 and day 20. One line had never come close. The board was quiet for a time. Velamaris spoke first. What does it currently consider its situation to be? We have no direct communication, I said, based on behavioral indicators. The schedule maintenance, the continued mathematical development, the measured physical exercise routine, it began on day 6. Our best assessment is that it has accepted the situation as indefinite and is managing. Accordingly. Managing, she repeated. It built a life in there, I said. A structured one. Voluntarily, without instruction. Another silence. Then the board member from classification, a senior analyst named Dorothy Kale, who had not spoken since the briefing began, said, Threat assessment on file lists the subject as Category 7. Standard sapient detention risk. Standard psychological fragility assumptions. Yes, I said, amend it. I amended it that afternoon. Category 3, elevated durability, psychological. The notation in the remarks field ran to 4 paragraphs, which is unusual for a classification amendment. I included the dissolution scores, the mathematical notation, the voluntary schedule, the 11-day wall, and what happened when this particular subject met it. What happened, in clinical terms, was nothing. The wall simply was not there for them. They had walked through the space where it should have been and kept going. Unremarkable, unhurried, the way you continue walking when you expected a step that was not there. A brief adjustment, not a fall. That evening I watched the last feed segment of the day. The human was finishing its notation, adding 3 new lines of symbol to the southern wall. When it finished, it set the tray edge down, rolled its shoulders once, and lay back in its designated rest position. It was on its 48th day of complete isolation, in a cell with nothing but its own mind, and it looked, for all the instruments I had running, like someone at the end of an ordinary afternoon. I closed the amendment file and logged it to the central record. Outside cell 14, the notation continued to grow.